Hello again, everyone. So I'll just quickly do acknowledgement of country. The University of Queensland acknowledges the traditional owners and their custodians of the lands in which we meet. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to the country. We recognise their valuable contributions to Australian and global society. Thank you, Matthijs. We're looking forward to your presentation.
Good morning, everyone, and thanks for coming to my talk. And also thank you to Matthijs Hollanders for inviting me, who's not here. And thanks for that. No, just kidding. So my name is Matt Hollanders. I just been doing a postdoc for the past 2 years or so, and during my PhD I got obsessed with stats. So I'm going to be talking to you guys about a few statistical approaches that I and others have used to tackle data sets. So a bit more of a background slide. I did a PhD in disease ecology from 2019 to 2023 at SCU in northern New South Wales. Before that, I was living in the Netherlands where I studied biology. Since then, I've been working as a postdoc with UC, and I'm about to start another postdoc with the University of Newcastle, where I'm going to be working on a bunch of camera trap data. And in 2022, I started Quantecol, which is a statistical consultancy where I help people analyze their data, or ideally with their study design. So, of course, ideally, you know how you're going to analyze your data to answer the question that you're going to have. And you can, if you get it right from the start, you can save a lot of time and effort as well. So I just wanted to start this presentation with this paper from a sociological journal, which is very simple. Their question is, what is your estimate? And the authors define the estimate as the target quantity, or what you're actually estimating with a statistical analysis. And when you have a question, you need to define your estimate so that you are actually answering the question that you think you're asking. So the simplest example I can think of is a t-test, for instance. Let's say you want to know the average difference in weight between eastern brown snakes and red-bellied black snakes, for instance. What you might do is you're going to collect a bunch of adult snakes, you're going to weigh them, and then you're going to do a t-test to estimate the difference in the means of their weight, and that becomes your estimate. So the more complex the statistical analysis gets, the harder it is to actually define this quantity, but it's critical that you do it, because otherwise, like I said before, you're not answering the question that you think you've asked. So the first example where this definitely came up was diseased frogs that I was studying during my PhD. So some brief background, I'm sure a lot of you are familiar with it, is a pathogenic frog fungus, the abbreviation is BD, caused a mega disease around the world with, I think the latest estimates are that up to 500 species were affected. I think 100 went extinct. Australia was pretty heavily affected. It most likely came in through the port of Brisbane, and everywhere it went, basically the species that were very susceptible went extinct within a few years. Others are still hanging on by a thread. And then there's other ones that probably initially declined and seem to have recovered since. So one of those, the one that I was focusing on, was glazed barred frog, which is endemic to the subtropical rainforests of northern New South Wales and southeast Queensland. I say probably declined because there really wasn't that much data from initially. Anecdotally, they were common in the 70s. Which, spoiler alert, I can believe, because in many places they're very common now. And then it seems like from the late 90s and through the early 2000s, a bunch of populations recovered. But the question remains, how are they affected by chytrid now? So the main part of my PhD was to investigate the effect of chytrid on these frogs. And the method I used for that was multi-state mark recapture. And we were identifying the disease in the frogs using swabs. So briefly, multi-state mark recapture is like any other mark recapture. You go and survey animals. When you catch new ones, you give them an identifying mark. And if you ever recapture them, you'll be like, hey, I found this one before, and you know that you saw it again. And any new ones, you give a mark. So what this yields is essentially a time series for each individual that is highly imperfectly observed. So if you had perfect recapture, you basically just have any medical study basically where you go and follow up on individuals over time. Here, because we can't check in with the individuals anytime we want to, we have to accommodate this imperfect detection. So how multi-state mark recapture models differ, we're not just estimating survival between occasions, we're estimating survival for different states, which here I defined as uninfected and infected frogs. Additionally, so you have these frogs that are either uninfected or infected, and they can also transition between occasions because you're not infected all the time. So between occasions, you can gain an infection or clear an infection. So we were particularly interested in relating that to, for instance, temperature, because that was believed to be a big driver of infection dynamics, where the highest prevalences were often observed in winter. So this is one of the streams where they occur, which are quite scenic. Turns out we're pretty bad at detecting BD, especially in this species. So another study I was doing the exact same thing with mark recapture, but on juveniles. So instead of giving them pit tags, I was photographing them in situ, and I was able to use the markings on their back to identify individuals, which worked really well. This was a high frequency study, and I was going like every week, and it turns out I had really high detection probabilities. So I was getting some pretty good time series on individuals, but once I had analyzed the swabs with qPCR, I became a little bit sus on the data because, for instance, this time series, this is a hypothetical time series, but some like this were observed, was basically saying in the first occasion, it was detected as infected, so state 2, then we didn't infect it, then it was detected again and it was still infected. Then suddenly when we detected it, it wasn't infected, and then it was infected again. So although it's possible that that individual cleared its infection 1 week and regained it the next, it's also likely that those would have been false negatives. So we actually failed to pick up the disease. So if you're not assigning your individuals correctly as infected or uninfected, it very much affects your analysis because you're not necessarily estimating the survival of infected or uninfected frogs. And especially the transition rates, for instance, in this example, if you don't account for the fact that going to uninfected might be a false negative, then essentially this individual gained and cleared an infection over 2 weeks, which may not have happened. So the way I ended up solving this is the swabbing process can be thought of as an occupancy process, where if you collect multiple swabs from an individual, you could treat those as repeat surveys of a site. So let's pull this up. I don't know how many of you are familiar with occupancy models, but here this variable, you see my mouse? Yeah. This variable Z is whether or not a site is occupied by a species, or in this case, whether or not the individual is infected with BD. And then Y are our repeat samples, and they are infected with a detection probability P, given that Z equals 1. So if you're not infected, you can't get a positive swab. So this ignores false positives. But if you are infected, you can still get negative swabs. So what this meant is that we basically needed to collect multiple swabs from the same individual. Or a nicer way to do it is if you have robust design sampling, which is when, let's say you sample every month and then you do rapid surveys within a short amount of time where you assume the population is closed. So let's say we do 3 nights in a row. Some individuals are detected on 1, some on 2, some on 3 of those nights. And every time you capture the frog, you take a new swab. So one of the assumptions there is that our swabs are independent samples of the disease on this individual. But this was a nice example, or if you can't really correct for this after the fact. If you don't collect the multiple swabs to begin with, it's really hard to correct for. Like you can use things like informative priors, but the pathogen detection process also depends on your loads. So I'll get to that in the next slide. The question is, or one of the overarching questions is, does it actually matter to account for this, or do you just do this because you like stats, basically? So it turns out in our system, we only had, where I estimated, a 60% probability of detecting BD on frogs. You might be wondering why it's so low. I think it's related to the fact that these frogs had very low loads. So they may have become resistant to the fungus, which means that when they have the fungus, they don't carry very high loads. Our swarming protocol was not very good at picking up these low loads, therefore you miss a bunch of infections. So as I mentioned before, particularly not accounting for these low detection probabilities of the disease, you inflate the rate of change. So here you needed to have an uninfection and an infection at the same time. And importantly as well, the estimate, the target quantity that we're estimating, if we're looking at the mortality rates of uninfected and infected frogs, if you don't account for the uncertainty in the disease detection, you're actually estimating mortality rates of individual frogs that return to positive swab, which is not the same as an infected frog. So in this case, if you relate it back to the defining your estimate is not what you think it is. So here I compared the output of the model. So the gray is in multi-state, and these ones here are the rates of gaining and clearing infection. So they were about 5 times higher estimated if you don't account for the fact that that some of the samples are you're not picking up the infection. And it turns out with this species, the mortality rates were very similar between infected and uninfected frogs. But at the very least, I felt more confident that what I was actually estimating in my model was the target quantity, which was the difference in mortality rates of uninfected and infected frogs. So this is sort of unrelated, but some of these mark recapture models can go very slow, and not all implementations are super efficient. So I've been working on the side on making, writing some really efficient functions to fit these models in STEM, which is a probabilistic programming language that's my tool of choice these days. So if anyone is interested in having a look at that or has big data sets that they're struggling to fit in a reasonable amount of time, definitely reach out. So the next example I wanted to discuss was related to koalas and their bellows. So at the moment, a very popular monitoring approach for koalas is through song meter arrays. And they're really good because they're basically super cheap, and you can cover a big amount of area for a pretty low cost. And these song meters, they run continuously, usually at night. And so you have, let's say, 12 hours of recording to get through. So I haven't done any of the machine learning stuff, but there are good algorithms. It's very simple now to just give this to a recognizer, and they give you back a spreadsheet of a timestamp or of a bunch of timestamps, or sometimes with a koala detection. So most approaches that I've seen that analyze these type of song meter data just end up collapsing all of the data of a night to a 1 or a 0, or whether or not we had a detection. And this is understandable because the methods that we're using, particularly or usually occupancy models, they were really designed for discrete surveys. So you can imagine if you go and manually go to 100 sites and you're looking for koalas, you're not going to sit there from sunset to sunrise and you're going to count every timestamp of a koala. You're going to go for half an hour. You're either going to pick it up or you're not, or better, you record the waiting time until you get your first detection, and you have this sort of time to event stuff. So it's understandable why people aren't using more of the data. But there is this whole call rate component on these song meters that are usually getting ignored. So a very basic model to account for those call rates is to not just have 1s and 0s, but to actually model the number of detections on a given night. So some sites might have more detections very often, and that way a 1 and a 0 is a 1 and a 0. Like if you have 1 detection, that's the same data point as a song meter that has 10,000 calls in a night. It's just it all gets collapsed to a 1 and a 0. So you can model these number of detections as for instance, a Poisson distribution, which is a distribution for count data, like a negative binomial or whatever. Additionally, you can account for zero inflation, because again, if you actually start looking at this data, a lot of the time sites will have nothing for days, and then suddenly there's 2 or 3 days where you have calls, and it's like, okay, maybe a koala moved in, but you want to sort of account for that in your data, that maybe some nights the site's not occupied, and other nights it is occupied. So great, we have this call rate, you've made the statistician happy, but can we actually do something with this call rate that we couldn't do with just 1s and 0s? So ideally, this call rate tells us something about abundance. So in hindsight, it's quite obvious that more calls probably means more koalas, but you can't get that information just from this data alone. You need to verify it with some type of other method. If you're happy to make that assumption, and modeling and statistics is all about making assumptions and what assumptions you're happy to make, that's great, but we wanted to know how does this actually relate to other measures of abundance. So what I would imagine is the gold standard of actually counting koalas is thermal drones. They work really well, you fly them over the forest, you see them at night, you can count them, great. So we did this for across 20 sites and 2 to 4 sites per night. And a naive, probably fine implementation of looking at that relationship is to model the number of bellows as a function of the drone counts that we observe. So when you do that, you get this nice straight line through a plot. It's pretty noisy, but me again, as the stats obsessed nerd, this doesn't really capture the structure that's present in the data, which is that sites were counted multiple times. They also had song meters multiple times, and the nights are also, some of these data points are from the same site on or across different nights. So you get this structure in the data that's not captured by this. So another way to approach this is a mixed effects model where essentially you fit you add a random intercept like this for a site, or a random intercept and a random slope for a site. But this actually gets you to a different estimate once again. This sort of answers within a site, when the counts go up, what is the expected change in our bellow rate, which you get this phenomenon in statistics called Simpson's paradox, which is that the overall trend can be like this, but when you start looking within groups, it's actually like this. So it's conceivable, for instance, that given a certain local abundance at a site, when you get more koalas, the call rate actually goes down, for instance, because they've already found each other, let's say. What if they're using vocalization to be like, hey, where are you, where are you? Suddenly they know where they are and they stop calling. So you might have, within a site, higher drone counts actually leads to fewer call rates. I made all of this stuff up, I don't know, but the model doesn't really capture what we're after. And as you can see here, we only had 2 to 4 replications within a site. So we're essentially trying to fit these different slopes for very few data points, which doesn't really capture what we want to do. So in this example, in hindsight, again, it was pretty obvious to me, but it's funny how long it can take. A better way to approach this is to consider the counts and the bellow rate as a multivariate realization at the same site. And one way to do that is to, for instance, just model the counts with random site effects and a random survey effect. So what this will yield is an average expected drone count for a site, and you do the same thing for the bellows, an average expected vocalization rate for the count. And if you model those random side effects as a correlated bivariate random side effect, you actually get a correlation coefficient, which tells you on average, what is the correlation between drone counts at a site and bellow rates at a site. So this is nice because it captures the structure that's in the data, the fact that we have repeat measures at the level of sites and at the level of nights. And as you'll see, it sort of cuts through some of the noise that's in some of the data. So one of the outputs here, we had 23 sites. So the black points, I realize it's a little small, but the black points are observed drone counts on the left. And then the point intervals are site level estimates of the average drone count at these sites. And just as an aside, this is the first time I've analyzed real world data where the Poisson assumption actually held. There was no over dispersion in the data according to this model, which was cool because normally count data is all over the place. As you can see on the right, for instance, if you look at the second site, we had some of the highest bellow rates of any site here, and then 2 nights it was dead quiet. So as another little aside there, I noticed some weird things in the data, and there was one night in particular where 4 sites were surveyed with song meters. On these nights, every site had the highest counts of drone or on the drone flights, and not a single call was recorded from any of the song meters. So we were joking that the koalas might be doing a blood ritual or something like that. Either way, all of them were in the forest and none of them were making any noise. So anyway, back to this data, our call rates are very over dispersed, as you can see. But if you look at the correlation coefficient of the site level expected drone counts and bellow rates, they were very high. So this was roughly 80% correlation. And if we plot the bellow rates versus the expected drone counts here on the left, you can also visually see that this relation holds. So our estimate here is basically if you take an average, if you take many measurements across multiple nights of drone counts and bellow rates, are these measures correlated with each other? And our goal here was not to go from, hey, you recorded 10 koala calls, that probably means you've got, I don't know, 6 koalas there or something. It was just to be able to have some sort of justification for using variation in these call rates as a proxy for abundance. So now suddenly we can use these song meter data, estimate call rates and model them flexibly, whatever, as a function of nights or seasonal effects or temperature or wind speed or whatever. But if you add a site level random effect, you can still be like, hey, these sites have higher drone count or higher bellow rates, they've probably got more koalas in the system. And as you can see here on the right, there's these different underlying models that I used for the bellow rates. So the Poisson is the same thing that I used for the drone counts, but the data was very over dispersed. So typically when you have over dispersed call data, you use something like a negative binomial. But I want to briefly talk about this Hawkes process, given that we're talking about flexible modeling. So of course, koala calls don't occur in a vacuum. You can have the same individual that vocalizes multiple times in a short amount of time. Presumably, you can have individuals that are calling to each other. So one nice way to model this is with a Hawkes process, which is called a self-exciting point process. And I borrowed this figure here from this paper where I learned about Hawkes processes in the first place. The idea is you have this baseline call rate where it starts. And the way the model works, the moment you have a detection, the call rate peaks by some amount, which is a parameter to be estimated, after which it declines again. So if you have one call, the call rate goes up, it drops down slowly, and then you have another call rate and another call and another call. So it's this sort of self-exciting process that increases the underlying call rate, which then declines back to baseline. So this was a nice way to capture mechanistically, potentially some of the over dispersion that's present in the data. And nicely for me as a modeler, it turns out this model had the best predictive performance of all the models tested. And even the correlation coefficient of the site level call rate and drone counts went up when you accounted for this over dispersion. So it was a nice way to mechanistically describe some of the particular ecology underlying the over dispersion in the call rate, and it improved our estimates as well. The final example I want to talk about is the example of unidentifiable cats in mark recapture studies. So my colleague, Dr. McHugh, he was doing really backbreaking work in the border ranges. He had a bunch of camera trap arrays to study threatened macropods. And he was picking up a lot of cats in this system. And it's not surprising, it's one of the most diverse areas in Australia. It's volcanic soils, there's a ton of life everywhere. It's not surprising you have high densities of a predator here. But the current literature, which honestly probably wasn't very good at predicting out of sample, suggested that there were average cat densities in rainforests. So prior to this, it was a bit underappreciated how many cats were actually in our subtropical rainforest. So the average density was about a quarter per square kilometer. He was getting lots of cats. If you can identify individuals, that's great because you can use spatial capture, recapture methods. But the issue is most of the cats weren't identified. So black cats, we couldn't identify any of them. Tabby cats were pretty hard to identify because you don't always get them from the same side either. Even surprisingly, the black and white ones can be quite hard to identify because again, they're not really standing there showing you all their best sides and all that. So typical spatial capture recapture approach is you're estimating these individual activity centers based on where the detections are occurring or on which cameras basically the detections are occurring. One way to do this is to use a data augmentation approach in a Bayesian setting, at least, where you have these capture history of your identified cats, and then you add these all zero detection histories of cats that were never observed. Some of those cats actually existed and were never observed, and some of them don't actually exist, and the model figures it out. It's kind of black box magic, but it works. And normally these unidentified cats are discarded, so I don't remember exactly what our number of unidentified cats was, but I think it was close to half. So suddenly, you've got X number of cats and now you've got half to work with. So here, for instance, I'm skipping ahead a little bit because the model that Ben Augustine works, who is a real spatial recapture genius, it estimates the number of individuals that were actually detected on your camera. So you might have only manually identified 10, but the model spits out, hey, actually, it seems like you actually had this many individual cats detected. And Ben's solution was this random thinning approach, where you have these individuals that you consider the detections that come from identified cats as this binomial thinning process, where you've got your true detection history, which you can't observe because you don't know which ones are identified. The identified ones are a binomial subset of that with this random thinning parameter. And then his model is quite complex. You can't fit this in just any software because you have these custom MCMC samplers to account for the fact that you need to accommodate the constraint that the unidentified individuals is what remains from the truth, which is latent, and the identified individuals. But what the model lets you do is it allows you to use all of the detections. So again, using all of the data, which as a modeler, that for me at least, that is always just a holy grail. Like, can I use all the information that we collected, or do we need to take some subset of it to fit it into these pre-existing models? And in this case, the thinning parameter, so basically the probability of correctly identifying an individual depends on the pelage type. So here, we probably could have just fixed the black identification to 0 because you can't identify them. But you see how some of the cats that have different pelage types have very high probability of correctly identifying them, and some of them, like particularly tabby and black, very low probabilities of correctly identifying them. So the outcome here is that we had much higher cat densities than was previously believed, with about triple the national average. This number was pretty stable across seasons, but as you can see, there's still a lot of uncertainty here, which is just the nature of ecological data, because we're always accommodating imperfect detection, which is depressing at the same time as well, because if you look at any power studies or whatever from other fields, for instance, medical fields, if you look at the sample sizes they require and you relate that back to us, yeah, it gets a little bit depressing. But at least we were able to use all of this information to get to these densities. So I'm not sure I'm going for time, but I'm actually done talking. So either way. So yeah, please let me know if you have any questions or.
Thank you, Matthijs. Does anyone have any questions?
One in the back.
Thanks for giving a good talk. I have a question about the first project.
Yeah.
So you went out and you did the swaps, and then you get script about the error rate of your swaps. Is there a way to assess what kind of error rate would have what kind of effect on your analysis? Similar to like our analysis, where you sort of beforehand you can do some tests and see where you would fall, and then whether you should account for this in the model, or whether it's fine to just.
The first question, I would just assess it with simulations. Essentially, you just simulate fake data under certain parameters, like let's say 50% survival, 10% detection. Like normally you have some sort of idea what it's like, and then you just simulate data with different levels of false negatives and false positives, and analyze the data with a traditional model and see how far off it is. The second question, should you sort of correct for it or not? I think that you should just sort of always correct for it, because the output, if you fit the model that allows for false negatives and false positives, but if you don't have false negatives or false positives, your output is going to be the same. It doesn't actually make your estimates bad if you don't have detection issues. So that my argument there is like, it's like with an occupancy model, right? Like, well, actually, I think sometimes we don't need occupancy models. We don't need to account for imperfect detection. Like, it's kind of just been, it's imperfect detection or bust basically in ecology these days, which I think is also not always appropriate. But with the disease stuff, I think if you have high loads, you can pretty well assume you've got high detection probabilities of the disease. So I should have included that figure actually, but I swapped all the frogs at the stream, and Fleae, the ones that we were studying, had the lowest loads by far. So if we had been studying the other ones, we probably wouldn't have had nearly as many false negatives, because there's more BD to pick up on the frogs. So it very much depends on your system. The tricky thing is with this one is that ideally you have the robust design sampling, so you have the multiple secondary surveys within a short amount of time, which a lot of people are already doing. And one of the painful things is the study that we were doing on the adults that was going on for 4 years. We were doing robust design samplings. We were going every 6 weeks and then 2 or 3 nights consecutively, but we didn't collect repeat samples. So we were this close to actually having like exactly what you need to properly estimate this. But I didn't develop this model until after that data was already collected, and it was basically too late. And there is of course the criticism of, okay, if you swab it the night before and you swab it the next day, like, are we getting a representative sample from the same individual, or did you wipe all the fungus off, etc.? I think here you got to choose between one of two evils basically, because if you have any reason to believe that you're going to have false negatives or false positives in your data, not correcting for it is just essentially going to completely screw up your infection dynamics estimates. Like in our example, it was a 5-fold difference, which means that the other ones were garbage. They're absolute garbage. Can't use it.
Yeah, I've got a question about the disease. Well, it's very difficult to study disease dynamics, obviously, more in wild animals. So something I've been involved with recently about mammals is eastern-barred bandicoots, as an example. They die after 3 days of getting toxoplasma. So toxoplasma is a little bit like chytrid in that it's in the environment. So it's not just getting transferred from one to the next. So, but you never find it in the animals. If you go and look for seroconverted ones, they're not there. So it's more a cause for alarm if you can't find any on the animal. So this is a big problem with looking at disease dynamics. In a way, it's not surprising that you found no difference in the survival rate of the ones that had chytrid on their back or not, because they're living with it, not dying from it.
Yeah, exactly. Yeah.
So this is I don't know how you overcome this. Maybe looking at the chytrid in the stream rather than on the frog and doing it at that level.
It's if you can't. Like I said before, with marker recapture, like there are time series that we're imperfectly observing. What you're saying is the chance of detecting one between it contracting the disease and dying is so small. There's essentially going to be no information at all. And
it's a much higher chance if they are quite resilient. It's a very, very low chance if they die.
So they don't always. I don't
know if this is the case with chytrid. Maybe, I mean, it sounds like it would be. If there's some variation between individuals, some of them are resistant and some of them are not. So the ones that are not, you're not going to find them.
Yeah, so but with the bandicoots, do they always die the moment they contract infection?
There's zero conversion.
Yeah, so in that case, a multi-state model basically doesn't really work. You'd have to look at either, I don't know if there's populations that are naive and look at just essentially you can't observe the effect of the disease because you'll never find animals with the disease, but that doesn't mean it's not happening, of course. But in that case, yeah, mark recapture does, because also if you first capture an animal and it's infected, and if you never or if you never get any recaptures, or in your case, you'd never even find an infected animal. So there's no data basically to estimate any of the parameters associated with that infected state.
Yeah, this is the problem with trying to study disease.
Yeah, I mean, we're lucky in that sense with chytrid in that, well, chytrid apparently frogs have it for, or the ones that are very susceptible will have it for a long time. And they're asymptomatic until like a day or two before. So when you see a sick frog, it's on the verge of dying, but you can pick it up in the weeks beforehand. So chytrid is good in that sense, is you have some time to recapture individuals. And what I would say with you'd want to increase your frequency of surveying to try and capture some of that, but you were talking about 3 days. So at the, like how even that doesn't sound very feasible. So yeah, sometimes you just out of luck.
Yeah.
To use a certain method, right? To use multi-state marker recapture.
I have a question online from Bob Galway. Thanks, Matt. I was wondering whether you recommend a particular approach to flexible modelling. For example, in all these examples, you've employed a variety of estimates and models to improve the use of the data. Is there any method to finding and choosing these, or simply always looking out for better options and relying on your and your colleagues' knowledge?
I think working in a probabilistic programming language of some sort is good because they're modular in a sense. You can plug things into each other and you learn to think much more generatively about how your data came to be. So any type of flexible approach for none of these methods at this point, there's just an R package that you can use to basically fill out a function and get input. So yeah, the what I always try to do is I think of what do I want to know? What's my estimate? So for instance, with frogs, it's the difference in mortality rates between uninfected and infected frogs. What's my data and how do I get from my data to my estimate basically? So just to use an example with frogs is we collect swabs and every swab goes through PCR multiple times. So like my raw data for this is the outcome of multiple PCR runs of multiple swabs. So the multiple PCR runs can be modeled like an occupancy model to tell me, is my swab infected? Because you can also get false positives in your or false negatives in your qPCR. Then you estimate your swab infection, and you have multiple swabs, which will tell you, is my individual infected, yes or no? And then that plugs back into the multi-state marker recapture to see what's happening to these infected individuals. So overall, I think the approach is to work in probabilistic programming languages, get familiar with how to write models and think of plugging different things into each other to get from your data to what you want to know. And having said that, Ben's approach with this random thinning stuff actually requires not just writing code in a language. He wrote custom MCMC samplers to accommodate certain things, which is a level too far for me as well. So yeah, but to begin with, and I think it's helped me a lot to work in these languages because it just, you see the relationship between different things, how things are often more similar than they're different types of models, and just helps you think more generatively about how your data may have arisen. Hopefully that sort of answered the question.
I have another question online from Carly. Thanks for the talk. When examining a large data set, how do you deal with any potential outliers? Are they always excluded, or are there cases where you would keep them in a data set?
I don't exclude outliers unless I think that the data is wrong. So that, I mean, and this even, and I know a lot of people experience with stats make this mistake, is we often don't look enough at our raw data. So that's a great way to pick up outliers. But for instance, I don't delete outliers unless I think that there is an issue. I just try and accommodate the outliers with a model. If the outliers can only be understood as outliers in the context of the model that you're using, because usually it's according to some distribution that you're using, that point is an outlier, that does not mean that it's actually an outlier. Like a data point is a data point if you are willing to assume that the data point is correct. So for instance, with these koala calls, those were definitely outliers that we had these sites where the highest drone counts on those nights didn't have a single koala vocalization. So my first step is let's actually listen to those recordings and make sure that it's correct, because I'm sus about whether that data matches. But when it turned out that that was correct, then I have to just assume that that data point is correct. Otherwise, where do I draw the line between picking and choosing data points? So yeah, outliers, keep them unless they are wrong.
Great, thank you. Oh, you sorry.
Is there a way with the koala to the bellow rate to basically apply a like polling probability from particular individuals so that's accounting for individuals that were there but didn't actually bellow? Is that the thing they're doing?
I mean, a big unknown here, which is I'd say another holy grail, is actually identifying individuals with their calls, because suddenly individual-based models like mark recapture, spatial mark recapture, all that stuff, they are powerful because you're following individuals over time. Whereas things like occupancy models and also end mixture models, stuff like that, you're counting individuals, but you're not identifying individuals. So the moment you can identify calls to certain individuals, that's great. As far as I know, the method for that doesn't exist yet. And also I haven't been doing any of the machine learning stuff. I don't know to what extent you can just write this up in Python now and it tells you, you know, this is Ben or whatever. But all we had was just a koala call. And that was one of the things, like we initially, before we did the drone count stuff as well, like I wasn't comfortable making the assumption that a call rate meant more koalas, because you can conceive of the idea that when there's no koalas, one koala really wants another koala and is calling non-stop. Like I said, without collecting auxiliary data, there was no way of knowing that without making untestable assumptions, basically. But yeah, that would be great, absolutely, because that will change it again, the whole thing, if we can start identifying individuals.
Yeah, but that's what something we do small apes in givens. It's a calling probability. I don't fully understand all of it, but it helps with the territorial different groups and things.
Yeah. I don't think we're there yet with koalas. At least we're not.
Just sticking on the koalas, Matt. So what was the implication of that work? So you found this relationship between bellow rate and count.
Yeah.
And so you can be confident that bellow rate has is an indicator of fungus. Is that all that they wanted to know? What did they?
It's all I wanted to know. So the thing is with these song meter arrays are really good for long-term monitoring, because you select a site, you presumably buy the song meters once a year, you put fresh batteries in them, run them till the batteries are dead. Like, it's just a great way to efficiently get a bunch of data. Normally with occupancy, all you're interested in, like, hey, is the site occupied or not? If you're looking at changes in occupancy from year to year, I think I put that as a bullet point in the slides, but I can say it out loud. If you choose to interpret that literally, going from occupied to unoccupied means a local extinction event, right? Like, there were koalas there before, there aren't any now, which is not a very powerful way to look at trends in species. So I know that we don't have that much data yet, like this was literally 20 sites, but I feel like the correlation was really strong that we can actually start looking at variation in the call rate from year to year. So with long-term monitoring, if now you're focusing on how that call rate changes, conditional on a site being occupied, you can look at trends in call rates, and if you're still after 5 or 10 years willing to make the assumption, or now, you know, hopefully tested it with some more data that relationship still holds, you can actually now use these song meter arrays to monitor a proxy for abundance, right? Like, people are so obsessed with abundance, they want to know, like, no, how many are there? Whereas I think more like, okay, we've got these song meters. Do we want to know exactly how many there are, or do we want to know the trend or the time series of how many we think there are using certain proxies? So to me, I think it's made the song meter stuff a lot more powerful, because it's gone beyond occupancy to some measure of how many koalas there are.
Yeah, I agree. I was just thinking about whether they would And whether, I wanted to ask whether they are pursuing that mixed design going forward to ensure that that relationship
Yeah, I mean,
does hold or erode under particular conditions.
yeah, because I mean, as like, I just find animals so weird because one year they're doing one thing and the next year you're just finding something completely different. Like you find that with going out probing or looking for snakes, like things just don't, the relationship doesn't maintain, right? So yeah, I would, especially given that we're talking about thousands of song meters here and heaps of money and it's koala money, so it's disposable, you know. But I think they should just keep doing this at different sites, as long as you want to ideally pair it so that some of the sites on the nights that they're running, you're also flying drones over, which I think is valuable data anyway, because the drones are great, like because with these, you see how close there's very little variance within the actual count of the sites, which tells me like, hey, this is actually a great way to measure how many koalas are there because it's so consistent from night to night. So yeah, but in terms of like a large scale monitoring thing, I don't think it's feasible to fly drones over all these places all the time. But yeah, to keep verifying it, because it seems like a small price to pay, given how much you can get out of using the call rate as a proxy for abundance.
Can I ask another one?
Yes.
Just on the cat, so yeah, you had a huge error in,
Yeah.
understandably in assessment. Was there any habitat component in here, and with that, and you might be able to answer this Tuesday in terms of what cats do. In the rainforest, is there any variation you think anywhere in cat abundance? With habitat features, ridges?
No,
gullies?
we didn't look at that.
Yeah.
There was some talk of apparently according to the mapping, there's some like cool temperate and some subtropical rainforest. But you've been to border ranges, I think, and it was just in that entire Brindle Creek catchment, like it all looks very similar, so it wasn't, no, we didn't look at ridges versus creeks or anything like that. Yeah, lots of cats.
Yeah, amazing.
Any more questions? No?
Thanks a lot, everyone.
Yeah, thank you.
Thank you.